And our first speaker is Michael Snyder, and he is the Stanford Ashman Professor and Chair of Genetics and the Director of the Center of Genomics and Personalized Medicine. Dr. Snyder received his PhD training at the California Institute of Technology and carried out postdoctoral training at Stanford University. He's a leader in the field of functional genomics and proteomics. His laboratory study was the first to perform a large-scale functional genomics project in any organism and has launched many technologies in genomics and proteomics. These, including the development of proteomics chips, high-resolution tiling arrays for the entire human genome, methods for global mapping of transcription factor binding sites, paired-end sequencing for mapping and structural variation in eukaryotes and RNA-seq. These technologies have been used for characterizing genomes, proteomes, and regulatory networks, and some of his seminal findings include the discovery that much more of the human genome is transcribed and contains regulatory information that was previously not appreciated. And a high discovery of transcription factor binding occurs between and within species. He's a co-founder of several biotechnology companies, including Protometrics, which is now part of Life Technologies, Afomics, now part of Illumina, Acellix, and Personalis, and he's presently serves on a board of a number of other companies. Welcome.
Okay, great. Well, thanks very much for including me in this symposium. It really is a terrific lineup. You've been hearing about genomics and integrating personalized medicine in cases where people have disease or ailments. And really what I want to do is just give you a perhaps more limited perspective in some ways, because I'm only going to talk mostly about one person, but the idea of integrating genomics and personalized medicine into an otherwise healthy individual, and what can that do for you and your potential health care. So I'll start with the obvious. This is something we all know, where really our health is the product of both our genetic information and what we've been exposed to, whether it's pathogens, the food we eat, or what have you. And really, I'd say a goal of all of us is to try and understand this in incredible genetic and molecular detail so that we can become completely predictive. When a child is born, you would like to know at the individual base level what every variant can do or is capable of doing when exposed in a certain environment. And I would argue, again, that's a collective goal of all of this. Now, I think we can get at this first part here, as you've been hearing from all the next gen. Maybe I'll move this up. Oops, there goes my joke slide. From all the next-gen sequencing that's been happening, basically the cost of genome sequencing has been dropping dramatically. We now pay about $3,500 to get a human genome sequenced, although it costs much, much more than that to clinically analyze it, probably somewhere on the order of about 5 to 10 times more in our hands. So with the cost of genome sequencing, the number of genomes that's getting sequenced is in fact skyrocketing. Starting from a very limited number, only 16 or so were done in 2009. And Nature estimated 30,000 would be done last year. I don't know if that number was attained or not, but certainly many thousands of genomes did get sequenced. And it's been estimated that 300,000 human genomes will get sequenced this year. So really the question's not shifting from whether you can get your genome sequenced, but do you want to get your genome sequenced? And if you do, what kinds of things will you learn from this? So just out of curiosity, how many people in here would like to get their genome sequenced? Okay, a fair number. How many would not want to get their genome sequenced? Okay, about 50-50, maybe a little more on the nots here. How many don't know? Okay, not so many. Wow, very definitive group. Okay. We'll see what happens by the end of this talk. I'm not trying to change you from one camp to the other, but I'll at least relate to you my personal genome experience and what I've learned by going through this. And again, one thing I do like to do is put this in perspective. So it really does cost, as I say, $3,500 to get a human genome sequence. And I was out shopping for hot tubs the other day, and I realized that, in fact, you can get 2 human genome sequences for the cost of a hot tub. So if you think about value there, you can decide which one is more valuable to you. And which one might be more beneficial to your health, yes. We'll see at the end of this talk if you feel that way. All right, so where is genome sequencing going to have impact? And clearly some of these areas are no-brainer. I would argue the first two. One is in understanding and treating human disease, these days, sequencing late-stage cancer patients is a very reasonable thing to do. We certainly do that, as do many other groups, to try and identify mutated genes. Cancer obviously is a genetic disease, as you've heard already, both in terms of predisposition and somatic changes. And the goal is to try and find variants that are in fact actionable. And quite frankly, from every genome we've done, which isn't a huge number, it's on the order of tens or so, one can always find one mutation, at least one mutation, that is potentially actionable with drugs that are either out there already or in early stages. So this is certainly a reasonable avenue. The other thing we can do, as Heidi mentioned earlier, is we sequence what we call mystery diseases, where you might see a child with an ailment or a disease of some sort, and it's very difficult to figure out what's going on, and by sequencing the family, sometimes, but certainly not the majority of cases, sometimes you can find potentially likely causative mutations for that. In our case, I would say we haven't found anything actionable yet, but there have been other instances where people have. The other area where genome sequencing is having enormous impact is in cataloging human variation, which is also important for understanding disease. But the 1,000 Genome Project now aims to sequence 10,000 human genomes, and they've done somewhere on the order of 2,500 at various states so far. And again, the goal there is to catalog human variation. But the area one thing that keeps popping up is, what about personal genome sequence? What would happen if you're a healthy person? Will sequencing your genome be valuable for implicating somehow or using somehow in managing your health? And this is something a lot of people think about, and most people think that, well, someday it will be valuable. And in fact, at the last ASHG meeting, that seemed to be a recurrent theme over and over again. Someday, human genome sequencing will be valuable for managing your health care. And then the question is, what day is that? And I would argue that that day may be sooner than you think, at least in some cases. And I'll be the first to say that we can't interpret everything going on with the human genome, but you can, in fact, learn some things from the information that's there. So one way of thinking about this is this, that you would be getting your genome sequenced, perhaps, and then together with a physician, try and make some useful information in terms of predicting risk. Probably wouldn't do that with a physician, do that with a genomicist. Perhaps diagnose, monitoring, and treating diseases based on this information. And minimally, I would argue that getting your genome sequenced ultimately should be valuable for understanding disease states. Now, you wouldn't do this on your own necessarily. You would do it with both your family history and a medical exam. And presently, if you get a blood test, typically they measure a few things, maybe on the order of 15 or so would be a pretty typical blood test. Maybe if you're sick, they'll measure a few more things. But really, the number is low. And thinking about today's technology, we were thinking that this seemed ridiculously low, that with the advent of deep transcriptomic profiling and proteomics profiling, metabolomics profile, one should really be measuring thousands, if not tens of thousands, of molecules in any fluid you might want to analyze, so you can get a much clearer picture of what might be going on in a particular physiological state. So what we've been thinking about a lot, and this really, I'd say, came to fruition. We've been thinking about it for some time, but it came to fruition with my move to Stanford in 2009, so almost 3 years ago. And the idea is to set up personal omics profiling for basically understanding human disease as well as managing health. And we obviously are big believers that your genome should be valuable for this. So the idea was to get your genome as well as a series of other omics profiles. And these are the profiles that we are either doing or have done. And one is the epigenome, which we're in the process of doing, so DNA methylation patterns. Transcriptome, where we measure mRNA and microRNAs. Proteome, and in our case, that's defined as what you can profile using mass spectrometry. Again, for the aficionados, that's using a Velos Orbitrap, where we consistently follow about 4,000 things at different time points, so 7,000 or so overall. This does not find everything, so we have specific tests looking at about 50 cytokines using ELISA-type assays, high-throughput assays there. We also follow metabolome, again using mass spectrometry. We follow 5,000 or so peaks, a little more than that, of which 1,000 we can identify. So most of these peaks you actually can't identify. That's a limit to metabolomics. We're also big believers in following the autoantibody profile. That is what antibodies you have that react with various antigens. We think that your immune system is a great monitor of disease states, and that by following your profile, this could be ultimately very valuable. We've done some studies along these lines. And an area we haven't incorporated, but I think will be very important, is incorporating microbiome information. So the goal, again, is to put all of these together into a personal omics profile and try and monitor physiological states. So when we started this, we wanted to find an individual who would be a guinea pig for this, so to speak. And we wanted someone having done many studies of the sort before, where you always run out of precious material sooner than you think. We wanted someone whose material we could get to rather quickly, and also in plenty abundance. And also there's this whole issue brewing at the time, remember 2009, not many genome sequenced when we started this, of, well, you learn harmful information from genomes and somebody might learn something and sue you. Remember, in America, it's very different than perhaps Canada. And so there were all these kinds of concerns as well. So the idea was to pick someone where we could get lots of material and not worry about getting sued. So of course that turned into me. Okay. So basically when we started this study, in fact, when I first moved to Stanford, I did save a blood sample, actually, when I first enrolled in a health plan there. But we launched a study on this particular day. It turns out I had a rhinovirus infection. It's just serendipitous when I got cleared for doing all this. So I had that's a common cold, so I had a rhinovirus infection. So we took that day and then several recovery days, 4 days later, 21 days later, and then periodically sampled over several months. Now I have 2 little kids and I pick up their infections quite regularly, so 9 months later, in fact, I got a very nasty respiratory syncytial virus infection and was out for several days, which, if you know me, that was quite a nasty cold, because I typically go into work and infect everyone else when I'm sick. So in fact, but this one we were all over. As soon as I started getting congested, I took a sample, then the next day, 2 days later, you can see very dense profiling, then more healthy states. So we've now been doing this for, I think we're at about day 730 or something like that. So basically we have 41 different time points. I've been through 5 viral infections. The 5th one, unfortunately, I was traveling, so we didn't get good sampling then. But for 4 of them, we have very dense profiling during the infectious time, and then less frequent sampling during healthy states. And the idea, again, was to see what we could learn from omics profiling, personal omics profiling from the study. So we started by sequencing my genome because at the time there were, and still are, several competing platforms. How good is genome sequencing? Heidi touched on this. She does a lot of Sanger sequencing to fill in missing blanks. We actually wanted to evaluate the technology, so we in fact did Illumina, very deep paired end sequencing, as well as complete genomics, also very deep. And we also sequenced my genome with solid and such. And at the end of the day, we discovered, even with the best algorithms, we had to set up our own pipeline, which we've done, which you can access. We've set up a variant calling pipeline, and with single nucleotide variants, so single base changes, we find that there's about 89% concordance between these 2 leading technologies, which on one hand may sound pretty good, but when you're thinking about clinical genomes, that's not so great. There are hundreds of thousands of variants that are found by one platform and not another, and I can tell you there's several % of the human genome that's not touched at all by other platforms, by any of the platforms. And so we spent quite a bit of time trying to understand what's right, what's not right, how to improve all of this. And the bottom line is I can tell you that the overlapping variants that are found are pretty much always right, we think, by Sanger sequencing and a variety of different criteria. The complete genomics variants are mostly right based on Sanger sequencing and other means. And the Illumina variants are less right overall. They have a higher false positive set in there. Yeah, so there are more errors there, false positives and such. And we know that a lot of these platform-specific variants are in repetitive regions, but they're not just in regions you don't care about. Some of them are in regions associated with human disease. Tool Butte has assembled a database called VeriMed. I'll talk about that a little more in a bit. And basically, some of these variants are disease-associated SNP, including one that's quite relevant to my particular health situation. So, in fact, there are useful variants in here. The other thing we did was we were evaluating technologies at the time, so we, in fact, sequenced my exome with all the leading technologies, with Nimblegen, Illumina, and Agilent, and they all work pretty well. It's just what they cover differs a bit, but they all do perform quite equally. One of the interesting things is when we compared our exome sequencing with whole genome sequencing, exome sequencing is done at about 80x as we've defined it here, 45x for the whole genome sequencing. We actually found quite a few variants with exome sequencing that were missed with whole genome sequencing, which surprised me with the number that was found. And they are present. These variants are there in the whole genome sequence. Their frequency just isn't high enough to be confident calls. And so at the end of the day, we're big believers that you actually want to get your genome sequenced not just with one platform. So for any genome we care about, we sequence it with either Illumina plus complete, or we do an exome plus a whole genome sequencing, so we get very high confidence variants, and that does cover a lot of the genomic real estate that's there. And with this kind of analysis, at the end of the day, for my genome, we call it 3.3 million high confidence variants, things found with 2 different technologies. There's actually a large number of short indels, which most people are ignoring. And here the overlap is terrible between complete and Illumina. It's only about 20 to 25%, depends on the algorithm you use. So most variants are not called, and we've spent some time validating this, and neither is perfect by any means or even close to perfect. So there's about 800,000 detected by, or 900,000 by any one platform, but only 2,000 or so overlap. And then we've also called structural variants, something we work on quite a bit. Here we use different algorithms for calling these. There are many more than this, but this is how many high confidence ones we have. And again, there are many low confidence ones we keep an eye on as well. So point number 1 is that you don't just want to, go get your genome sequence is not as trivial as it sounds. There is, in fact, something to be said for looking at exactly what that sequence is and doing it right. And so we do spend quite a bit of time, as I say, for any genome we care about. We try and do this at very high accuracy, or as accurate as we can. Now, these variants I mentioned, they're not simply benign variants. Many of them are expressed and lie in genes, so about 31,000 lie in genes that are expressed. So we've done ridiculously deep RNA sequencing on my samples, 1 lane of high-seq for each sample. We're almost done all 41 samples we've collected so far, but from the first 20 or so, we collected 2.67 billion 100-base paired-end reads. Why do we do that? Well, we wanted to follow a heteroallele expression in editing, as you'll see in a minute. Of these variants, there are 31,000 of my variants are expressed, about 1,800 are non-synonymous, and some of the nonsense mutations are expressed. And from our mass spectrometry work, we know that a number of these are made as protein. They're not just completely sitting in the genome, not active. If we look at heteroallele expression, this is looking at the rhinovirus time course, so day 0 and then some of the subsequent days. It turns out if you follow your different alleles, this is my non-reference alleles, these are my reference alleles, there's a lot of genes that show differential expression. So remember, you have a paternal allele and a maternal allele, so you should never be talking about the 3 billion base genome, you should always be talking about the 6 billion base genome. Your alleles do behave differently, and I think that leads to some of your personal phenotypes. And if you think about it, humans have been selected largely by pathogens and such, so your different alleles are clearly going to have, we think, an important response in your pathogen sensitivity and resistance. And so by seeing in large numbers, there's hundreds of genes that show this differential allele expression during infectious states. And some of them are common between different infections and some are different. The other thing we've looked at is RNA editing, again, because by doing very, very deep RNA sequencing, we can call edits with very high confidence. We do find quite a few edits in genes. About 600 of these lead to amino acid changes affecting about 200 different genes. And again, some of these are expressed as protein. This is not quite as many edits as Vivian Chung reported about a year ago, which is, we think for technical reasons, we think she's probably overcalling her variants, but nonetheless, there are quite a few edits that are expressed that show up, and these now, we know some of them show up in different tissues. Okay. This is the thing you really care about when you get your genome sequence. What does it mean to you medically? So this is a pipeline you and Ashley, Atul Butte, and Russ Altman set up just before I arrived at Stanford for looking at Steve Quake's genome and then later John West's genome and now my genome. And we've added some contributions to this pipeline. So this is the pipeline, and people are now using variants of this pipeline in many different institutions. This pipeline makes certain assumptions, all of which are wrong, but it's a first pass to be able to interpret genomes. What we do is we initially take the whole list of variants, both high and low confidence, and start looking for those that are rare, because the assumption is the common ones you'd know about already. We do keep an eye on the common ones, as you'll see in a minute, so we don't completely discard them, but we mostly focus on these rare variants. We do look at non-coding sequences. I'll have more to say about that in a minute. Quite a few of them do affect mirrors. I have at least one that affects splice junctions. But most of our effort is spent on the protein coding region variants, because those are the ones we know how to interpret. We take these rare coding variants, and we look for those that are non-synonymous. I have 1,300 or so. Of these, about 234 are predicted to be damaging. using these programs, which are not perfect by any means. Again, don't get the right idea that these prediction methods are very good. They're okay. But anyway, I have 234 damaging variants. Of these, 51 are found in the OMAN database you heard about earlier, so they're associated with human disease. So I have 51 variants associated with human disease genes that are damaging mutations, and virtually all of these are heterozygous. As you might imagine, they're rare. This is my list of variants, I guess lesson number one is, if you're a worrier, you should not get your genome sequenced, okay? You will have a list just like this. Maybe the names will change a little bit, and some of the genes might look nastier than others. But we all have these lists, and this is just my rare variants. I have common variants for which I'm homozygous mutation. I'll mention, in fact, here's 2 down here, 1 here, 1 here. From this list, it actually is fairly benign for the various genomes we've looked at. Overall, I'd say it's benign. I had a nice positive control. I knew serendipitously I was heterozygous for alpha-1 trypsin inhibitor. Most of the other things aren't so problematic. By the way, invariably, some of you will see something interesting up here and will feel free to give me advice. I get thousands of free advice every day, it seems. Well, every week I do get definitely some suggestions. Anyway, by looking at this, there were a few things that did catch your eye. Number one was, in fact, this one right here. I have the exact same mutation. It's in a gene called telomerase. I have the exact same mutation as people with aplastic anemia, who are also heterozygous for the mutation. And if you read those studies, you would think I would have aplastic anemia. Well, we have measured my telomere length. They're normal by blots. They're a little bit smaller for my age group by FISH analysis. But nonetheless, they're not that much smaller, I think 9% or so. It's pretty minor. And not only that, we sequenced my mom's genome so that we could phase mine. That is, assign alleles to both my maternal and my paternal chromosome, and also to be able to interpret her genome. So we sequenced that, and sure enough, it turns out she has the exact same mutation. She's 84, going strong, and no sign of aplastic anemia. So I'm not so worried about that. There's nothing wrong with that study, by the way. That's a classic case of a contextual mutation. In the background that was being studied, if you looked at it, it looks like that mutation does cause aplastic anemia. Clearly, in our background, it's not as penetrant. Although I just heard Neil Reisch give a talk, and he's been looking at telomeres for a very, very large cohort. And it turns out your telomeres shorten over your lifespan. And for women, it turns out it's very slow, but for men, it starts a very steep slope, starts around age 50, and I'm 56 right now. We started this when I was 54. So in fact, I may have to worry about this, even though my mom didn't. So, oh well, she didn't have to anyway, she didn't know about it. But anyway, I was planning on measuring my telomeres the rest of my life, but now I'll probably watch them a little bit more closely after hearing Neil talk. So this is a case of, dynamic use of genomic information, I guess. Anyway, we'll keep an eye on that. Again, I'm not so overly worried about that. We did see a few other things that caught my eye. I knew I had high cholesterol going into this. I'm a great responder to statins, so again, that seemed to have all worked out fine. And this is likely one of the major causes of my high cholesterol. I have mutations in this gene. I can't remember. There are a lot of compound heterozygotes. I think that was the case in this one, meaning you have 2 different mutations. And this is important. When you get your genome, you want to get your genome phased, by the way. We did it the easy way by sequencing a family member for me, because you do have lots of what are called compound heterozygous mutations, meaning 2 different mutations in the same gene, but in compound heterozygous, they're all different alleles, so they can both be inactivating. And I have 139 damaging mutations that are compound heterozygous, probably more than I have for, in fact, homozygous mutations. So you want to capture all that information. That list did not look so bad, so I haven't presented up here. Okay. I also saw some mutations in these and other genes that have been associated with diabetes. Again, these are not obviously rare alleles. There are diseases rampant in the European background, so we did keep an eye on some of those, and so that was something that did pop up. The other thing we did was, in fact, integrate over all the information we had. So Atul Butte has assembled this database, VeryMed, where he's actually reread the literature. He's had a team of curators read the literature for over 5 years now, and basically look at all the GWAS and other genetic studies, and for each variant, calculate likelihood ratios. And then he set up a method for being able to integrate across all those alleles. Now, he'll hate me saying this, but it's sort of like 23andMe, because you're integrating across all your information and predicting disease risk. It's not perfect by any means, but it's something he's used to to predict disease risk based on this likelihood ratios. And second, what Heidi said, it turns out 10% of the information in the databases in our experience is wrong. They actually put the probe sequence in instead of the genome sequence when they're describing the alleles. So this is some of the issues you face if you're just dealing with the public databases. Anyway, this triangle represents a European male. The likelihood of getting a risk of one of these diseases, this is 0% chance, this is 100% chance. And orange means I have an increased likelihood of the disease by that magnitude, and blue means decreased likelihood. So a lot of this I knew already. I knew I was at higher risk. That's a small orange thing. I knew I was at high risk for coronary artery disease because everyone on my father's side has died of heart failure of some sort. So I sort of expected that. Here are the high triglycerides I mentioned before. You can see that shows up at a couple of places. Low obesity, all that made sense in my family, for my family history. But there were surprises I didn't necessarily know. One's fairly recent, this discovery on high risk for basal cell carcinoma, something I'll have to keep an eye on. But here's one that popped out of nowhere, type 2 diabetes. I was not aware of that in my family. I go from about a 24% risk to about a 47% risk, quite a big increase, as you can see from that line. So I wasn't aware of that running in my family, so I asked my mom about it, and she said that basically my grandfather, who lived to be 87, got high sugar late in life and just cut out desserts and everything was fine, so that's why it was never really mentioned. And you wouldn't call that necessarily highly penetrant anyway, because basically I have lots of aunts and uncles, and I've never heard about this in anybody else. So this, again, was surprising to me. But nonetheless, it prompted me to actually sign up for a glucose metabolism test. There's an expert at Stanford, Jerry Reven, who runs these tests. They're fairly deluxe tests. They take several hours to perform. And because of my busy schedule and their high demand, it took 5 months actually to get this test. But in the meantime, remember, we've been omics profiling me since the start of the study. And in fact, one of the things we've been following are my standard medical tests, including glucose, which shows up both in my metabolome and in my regular medical tests. And it didn't matter whether I was fasting or not, but it turns out my glucose level was running pretty normally through the entire first part of the study. But the time I went in for this glucose metabolism test happened to be that day there, and even before we started the test, after fasting, my glucose level was 127. And first of all, I should say, when I first went in for the study, the woman looked at me like, "You're crazy." In fact, it wasn't like you're crazy. She said, "There's no way you have diabetes. You don't have a family history." And I said, "Well, I know that, but my genome predicts something's wrong with my glucose metabolism. We have to do this test." It was literally the actual conversation. And so, she's like, "All right, yeah, we'll do this test." So she measures this, and it's 127. We were both surprised. So she repeats it, and sure enough, it's the same number. We do the test, and I am insulin sensitive. But because it came out pretty high, we did some, obviously, subsequent glucose measurements and did a better test for glucose levels, hemoglobin A1C, for those of you who follow this, and that's a pretty high number. At that point, I didn't really know much about diabetes because it wasn't running in my family. I hadn't really paid much attention to it. So I went to my general practitioner. That was about this day here. And she looked at me and said, "There's no way you have diabetes. Those guys got the tests all wrong because, I don't fit the normal phenotype here." And so she repeats the test. I'm sure they send the samples the exact same place, by the way. And once you know it, it comes out even higher. And in fact, my hemoglobin A1C was even higher, to the point where she said, "You're diabetic." Okay. So the bottom line is, in fact, my glucose level did get quite high after my genome had predicted it. And this is, again, not something I necessarily predicted. It was really quite a surprise. It's one of these things, yeah, my genome says this. I don't really believe it, but I'll kind of follow up on it anyway. So it was high, but it was sufficiently high, and this wasn't just for a few days. This actually wound up being high for several months. So on April 13th of last year, I completely changed my diet for something that was really terrible, which is eating lots of desserts and ice cream. I cut all that stuff out. I don't eat that anymore, so it'll be interesting to see what we have for lunch. But nonetheless, and I also increased my exercise. I was biking some, but I doubled my biking, started running. And it took a long time, but it took about 6 months, but my glucose levels did come down to normal without taking any drugs or anything. So, in fact, what I would argue is that my genome did predict a risk for a disease, which in fact I did get. And it's by doing this frequent profiling, and in fact, from the genomic information, certainly had me looking at the glucose, I was able to catch it early and then do something about it, managing early. And of course, that is the key to healthcare. You're going to hear talk after talk about people trying to treat disease, but really the ultimate goal is to treat things before you get the disease. And quite frankly, you might say, "Well, Mike, why didn't you do this before you got diabetes. Why didn't you do all those things? And that's true, I probably should have. But anyway, I did catch it early. And so again, that's the key, either catching a disease before it happens or its early stages before it becomes symptomatic in other ways. Okay, so that's one of the key lessons. My genome, in fact, was valuable to me. Now, the other thing you'll notice is that, in fact, my glucose shot way up right after this nasty RSV infection. In fact, I was out for several days, again, which is quite unusual. And I didn't take anything for it. I don't want to wreck the experiment. But in fact, it did shoot up after that. Now, viral infections have been associated with type 1 diabetes. Stress responses have been associated with type 2 diabetes, but there certainly aren't many studies that I can find out there that say much about viral infections and causes of type 2 diabetes. So I don't know that this is RSV specific. It might be more the stress response. But the other thing I've learned, and many of you probably know this already, is that diabetes is an incredibly heterogeneous disease, both in terms of its symptoms, in terms of insulin resistance, sensitivity, and drug responses. So in fact, really, it's probably hundreds of diseases. And this relates to the next part, which is we'd like to be able to subclassify this further, which we have not done, but an area that we're pursuing quite strongly. So the other thing we've been doing, as I say, is all this omics profiling, and I want to show you what we've learned from that. So starting with the cytokine profiling, these are the first 20 time points or so, starting from low glucose and ending in a high glucose states. These are not equal time points. These are the infectious stage. You can see my cytokines go ballistic during the various infections. I have allergies. We think that's responsible for some of these, although there is a fairly strong response that shows up in many signatures about 2 weeks after this RSV infection. Okay? We've looked at all of my omes, if you will, my transcriptome, proteome, and metabolome, both individually and collectively. And this was quite a bit of a challenge, but luckily I had a physicist join the lab named George Mias, who in fact took all this data, normalized it, figured out how to integrate it in a very useful fashion, and more importantly, figured out how to look for systematic changes in a longitudinal profile in which the sampling is uneven. That is a technical challenge. Okay, and he came up with a really terrific framework for doing this. And we wound up looking for two kinds of changes. We look for overall trends, if you will, in the data, meaning things that go down, things that go up over a long period of time. And we also look for spikes, dramatic changes that would occur at any particular time point. I'm going to show you the results for the integrative analysis because I think that's the most useful, but we did look at this again at individual transcriptome, proteome, and metabolome. So this is looking at all my components. It turns out we're following 40,000 different things at any given sample. Most of it's from the transcriptome because that's where you get most of our measurements, but we do get on the order of 4,000, a little bit over that for the proteome, and again, 1,000 known things for the metabolome. So this is looking for autocorrelation, general trends across a time point. So this is my respiratory syncytial virus starting here. Before that is my healthy low glucose state. Again, it goes up during these first time points and ends at a high glucose state later. And these are essentially the genes, proteins, and metabolites, or RNAs, proteins, and metabolites that go down. These are the ones that go up. And we systematically look for pathways that change during this time. And we find a lot of secretory type pathways go down. And interestingly, this is not cherry picking. These are picked out after Bonferroni correction. Again, which is actually a fairly stringent criteria when you're following 40,000 different things. In fact, we did see, anyway, one of the interesting things that came up near the top of the list was, in fact, my insulin pathway did show a decrease from that high glucose state to the low glucose one. As I say, the other thing we looked for were spikes in the data, and there was, as you might imagine, a huge spike during the viral infections because everything went kind of crazy then, not surprisingly. But interestingly, we found a very strong spike at about 2 weeks, again, slightly after that cytokine time point, it's the very next time point. We found a very strong spike of things that went up, and I'll show you in a minute things that went down. And I forgot to say back on this one, this is mostly a transcriptome signature. I'm only plotting RNA and protein. RNA is blue, protein is yellow. In this one, there's things that spiked up. Again, we could pick out pathways, and one of the things at the top of the list was glucose regulation of insulin secretion, spiked at 2 weeks. There's this whole pathway that showed this big charge up. There's some interesting metabolites that change as well, some of which have been associated with diabetes. There's also things that spike down at that exact same time point, and this is one on platelet plug formation and others. What's interesting about these is that some of these pathways you only picked up by doing the transcriptome and proteome. This is a mixed signature, and the confidence we got out of this was by following all these different components. If we looked at the transcriptome or proteome alone, we would have missed it. And I forgot to say this signature is mostly a proteome signature. So you are picking out different things. It's something we would all expect, but it's nice to see that by integrating across multiple components, you get a much clearer picture of things that are changing. It was quite interesting. This was not cherry-picked, that a lot of the things that changed right before my glucose spiked actually were implicated in either insulin signaling or glucose regulation. And one of the things that actually changed and went down here was, in fact, an insulin response pathway during that time point. The other thing we've looked at is, in fact, my autoantibody profiles. We've been doing this by probing an array of 9,000 human proteins, and more recently we've come up with an array that tiles the whole human proteome and trying to follow the response there. So for this first part, we just probed at the first 6 or so time points. Interestingly, near the top of the list, this is cherry picking, but we did find autoantibodies to an insulin receptor binding protein. I have no idea if this is general. Again, this is an N of 1. We do need to look at this more detail, but this is the kind of thing that at least can pop up in a study like this. Okay, the other thing you will learn from your genome sequence, I guarantee if you don't learn anything else, you will learn something about your drug sensitivity, your drug response. And this is a short list from a very long one of 5 fairly relevant to things. Hopefully I'll never need these, but I may. My sensitivity to warfarin, used in heart attacks and such, and there were several things implicated in diabetes. This is the first line drug you would use for diabetes, metformin. I hope I never need it, but if I do, I'll at least have some idea where to start out with the dosing. Okay. Also not shown here is actually some of the statins I should avoid based on side effects. So you do get information about your drug profile from your genome sequence. Okay, let's see how much time I have left. I want to just emphasize one thing that we have been doing is, let's see, probably have about, what do you want, 5 minutes? Sound good? Okay. So everything I've been talking about has been based on coding sequences. And it turns out we think for a variety of reasons that that's probably the least important regions in terms of what's the difference between people. And we know that from several different studies we've been doing, including some I'm about to show you. So again, the major difference between people we think is differences in the regulatory sequences that control expression of genes. And so we've been actually now curating all kinds of regulatory information to be able to use this to annotate personal genomes. And 2 big approaches we've been using is one, assembling a database that you can have access to. This pulls together information from systematic projects like the ENCODE project and other high throughput studies, but also manual curation by Mike Cherry's group, who has been manual annotating all those promoter bashing experiments people did in the 80s defining regulatory elements. The other thing we've been doing is mapping transcription factor binding in different people. We did an early study of this, and now we're doing a lot of this in quite a few different people with different marks. But in our first study, we mapped the binding sites of NF-kappa B across 10 different very diverse individuals. And you look for differences in binding with strong binding and weak binding and things like that. We also mapped POL2, which shows very strong differences. And there are quite a few variable binding sites for regulatory factors across different people. This is much, much higher than coding sequence variation. That's one of the reasons we think regulatory information is very, very important to incorporate in annotating genomes. We actually take this information, look for variable binding sites associated with particular genetic differences. So the first thing you do is you look for differences in the factor itself. So for NF-kappa B, we look for variations in the consensus sequence that lie under the peaks. And for a subset of these variable regions, we can find this. So if you change that G to an A, you will lose binding. Or likewise at C, there's a different location. People with the C bind and people with the A don't. So we can annotate some regions of the genome with this. That actually, it turns out, only annotates about 4% of the variable sites. Turns out it's not just NF-kappa B variation, it's partners of NF-kappa B, and we can tease this out informatically. I'm going to gloss over this, but we can find partners of NF-kappa B that also, whose sequence varies and affects NF-kappa B binding using some interesting informatic tests. And together, then, we can actually look at these variations, not only NF-kappa B, but partners of NF-kappa B, and annotate those as well. And the net result is to be able to integrate binding information with gene expression, which we can do by the expression information we get with these individuals, along with disease risk information going back to Varimed, looking at disease risk alleles and assigning it with these differences in binding. And the net result is at the end, we can again assign genetic variation with disease risk and biochemical events. And this is one for atherosclerosis. Actually, binding is the high risk allele, and naturally I have that one. Contributes to my higher risk for coronary artery disease. Okay, so where do I see this going? Well, I do believe for people who could get their genome sequenced, possibly at birth, not a decision made by them, no doubt. But the idea is that you might, I do envision a future where many people will want to see genome sequencing done at birth, certainly at a cost that it probably occurs now for PKU and other tests. In my world, you would also add other kinds of omics information. Profiling 20 things seems ridiculous to me when we should be profiling thousands. We don't know what the best 1,000 things to profile are, or thousands of things. That's why we're doing 40,000, or as many as we can. I do hope we can extract something out of that. And together with other omics information, we hope to be able to better manage human health. So the conclusions are personal genome sequencing is here. The medical interpretation can occur at some level, but it is extremely difficult. We spend about 200 hours, human person hours on a clinical genome. Genome sequencing can predict disease risk, which you can follow with other omics information. I really want to emphasize that with this more comprehensive profiling, you get a much clearer picture of what's going on in terms of physiological changes. I do think certainly an area we're pursuing is to add more regulatory information, genomic regulatory information into this. And this last concept is really one that I think many people have an issue with, which is that we profiled one person. A lot of people say, well, you only profiled one, you can't learn anything out of that. But I would argue for personalized medicine, this is exactly what you want. You want to know your altered state relative to your healthy state. I don't really care what my altered state is relative to everybody else in this audience, maybe a little bit. But the point is, really it's how my state alters in a disease condition relative to a healthy one that counts, and that's the essence of personalized medicine, and I'd love to see that all managed with a home test. That'll be a very controversial statement. So again, it's a different line of thinking when people are used to thinking, well, you have to analyze hundreds of thousands of people, like was done for diabetes, to start finding risk alleles. I could argue you can learn a heck of a lot from a single omics profiling experiment like we did for me, issues about hypotheses with viral infections causing disease and things of that nature. You can get a lot of information from a very detailed analysis of a single individual. These are the people who did the work: Rui Chen, George Mias, Hugo Lam, Jennifer Lee Putan, Liwa Jiang led this. It really started out with 2 people. More people just kind of volunteered to jump in on this project because it really was a lot of fun. For PIs, we had all these people. I mentioned a lot of them, but we also had help from markers, Jean-Kari Nadeau, Hua Tang. And I do want to thank my mom, who was a really good sport and helped in many different aspects of the study. Okay. These are the people who did the regulatory work: Maya Kosowski and Fabian Gruppert, and I mentioned Mike Cherry and Alan Boyle did this, helped in this work as well. So thanks very much. I did forget to ask how many want to get their genome sequenced now. How many don't? See, I had no effect. Okay.
All right.
Any questions?
Yeah, so we have time for maybe 1 or 2 questions.
Yeah, I did consent myself as part of an asthma study, which rhinovirus is an asthma. There's a connection there.
The second question is that if people sue you, say you are using public money to study yourself, what money?
Yeah, well, it turns out I used, those are the advantage of moving to Stanford. I used my setup funds for this. I was concerned about that. We did use tools that we had developed with NIH funds, but we used Stanford money just to avoid that issue. Although, don't get me wrong, this is a research project. This is not for the clinic. People always say, well, when's this going to be in the clinic? I don't know when it's going to be in the clinic, but I do think this is a research project. The goal is to extract useful information, make hypotheses, go out and do this further. And so our plan now is to do 250 pre-diabetics. About a third of them should convert over a 5-year period and follow different pathways of change. And again, the goal, if nothing else, is to understand disease states. And maybe we'll learn things that are of clinical utility as well.
I think that data about the glucose is really interesting. BMI, your central adiposity, insulin resistance, but it was acute, which we think of as type 1 diabetes, I guess, as a response to a pancreatic acute infection. Is that ever, I mean, I think that's a great study you're planning with the new pre-diabetics. Has that been seen before? There's like this acute rise?
Well, it's interesting how many people have walked up to me and say, we know that already. And I say, great, send me the PDF. I've yet to see it get a PDF. So it's one of these things maybe people have seen anecdotally, and I've gotten quite a few emails since we published this, which we did in March, of people who had a similar circumstance where something triggered. Now, again, I think it comes back to this issue. Diabetes is a heterogeneous disease. In my case, I don't fit the typical profile, which is why everybody's saying there's no way you have type 2 diabetes. But I think in my case, you know, our argument is that my genome had me predisposed with the viral infection, in fact, triggered the disease. Now, I didn't say, but in fact, when the disease reverted, I have lost 15 pounds since I changed my diet and increased exercise. Not intentionally, it came down. And in fact, my glucose levels tracked best with my BMI coming down. But again, I wouldn't hit the normal stereotype for a diabetic. Again, I presume I'm at risk because of my genome. Time for
Last
one
question.
Okay.
So the diabetic story was very, very interesting, but on the other hand, would you think that some of this information had to be masked? Because from a health perspective, I would be also following myself with an ultrasound for pancreatic cancer that might not even happen. So it will end up more saving on one side, but more spending on the other side for follow-up or for expecting some diseases that might or might not happen.
Yeah, well, this is why I think we need to get simple markers. This is why I enjoyed the previous talk for all these different things. And this is why we're profiling me so densely. I will die of something someday. If it's acute, the person after me will figure it out. If it's chronic, I hope to figure it out myself. But you can see that some markers should change. And I guess this is my philosophy. This is one reason we entered the study. I'm a believer there will be 1 billion human genome sequences out there, and certainly many millions in a very near future. Imagine a world where there's a few million human genome sequences linked to a few million electronic health records, linked to a few million of these longitudinal profiles. Medicine will never be the same. We will know so much more about biomarkers and health states than you can ever imagine. That's my view, and I'd like to see that become a reality. And we as a collective we, it's not a Mike Snyder we, because we're not going to do 1 million profiles, but that's what I would like to see happen. And then I would like to see these convert to very simple home tests where you can do a prick. I think it's so crazy. You prick your blood and you follow glucose. Why aren't we following thousands of things? That's what we're working on now. You can do that with today's technology. Why aren't we following 5,000 things with a little prick routinely? And that will give you a much clearer, earlier finding of what's going on. And for acute diseases, it's hard to make economic arguments. I mean, I hate to say it, but for pancreatic cancer, people die right away. That's not so expensive. But the chronic diseases, there's very good studies out there, and cardiovascular disease is the best one. It actually is economically justifiable to sequence people's genomes, because if they get a stroke, there'll be a long-term disability, and that's very expensive. So you can make economic arguments in somewhere. That's just completely erasing the whole value of life part of this equation too, which of course I think is an important part. But you see some of the issues. I do think you actually will save money in the long run. I would argue my catching diabetes early probably saved quite a bit of money as well. And people ask how much does it cost? Well, it's about $2,500 for a time point for me right now with my genome sequence. And that's mostly one lane of a high-seq, which is more than you really need. Okay, so those are some of my thoughts.
Great.